Hi, welcome back. Here we are onto the stomach, or as my son likes to say, the stummy. Stomach and tummy mixed together. Isn't that fun? All right, so hopefully you've gone through the anatomy of the stomach. You've gone through the layers and control of the digestive system, that first video. And now we're ready to dive into the details of the stomach. Again, I'm not going to go over all of the details of the anatomy of the stomach. I'm going to jump right into the physiology. First of all, taking a look at the stomach lining and the production of gastric juices, which is hydrochloric acid, intrinsic factor, pepsinogen, and lots of mucus. So the part of the stomach that is responsible for all of those secretions is the gastric glands, which are located deep in the mucosa of the stomach. So again, if you remember, we are going to look for those four layers. So here's our serosa. Here's our muscularis externa, there's our submucosa, and all of this is the mucosa. So those are those four layers of the stomach. And we'll notice one of the things that is different, and actually I need to correct my muscularis externa, because one of those modifications of the muscularis externa is the stomach actually has a third muscle layer. So not only do we have the circular and longitudinal layers, we also have what's called an oblique layer. So we have muscles going this way, we have muscles going this way, and we have muscles going this way. And structure and function, it makes sense because your stomach is basically like a big mixing bag. I like to call it the magic bullet. I bought a magic bullet a long time ago because the commercial says, oh, you can make all these wonderful food things. No, it makes really good smoothies, and that's about it. You can't chop things in a magic bullet. You smoothie things in a magic bullet. And that's what your stomach does. It basically takes all the food that you consume and it grinds it, or not grinds it, that's your teeth. It mashes it and mushes it and continually churns it until it turns into this smoothie-like consistency of a material called chyme. C-H-Y-M-E. I know it looks like chime, but it's pronounced with a k, chyme. So this is your mouth kind of starts that breaking down process with your teeth and your tongue and your cheeks and kind of mushing everything together into smaller chunks that you then swallow down your esophagus, lands in the stomach, and the stomach recognizes that, it starts churning and blends everything into this chyme. And while it's doing that, it's secreting these juices to mix in with the chyme to actually start some of the processes of digestion of proteins. All right. So let's take a look at one of these glands over here on the right. So we can see the parts and pieces. We have the gastric pit, which we saw on the histology slide in the first little mini lesson, the little kind of clear pockets that you saw on that slide. And then the rest of it is the gland. And the gland is composed of a couple different kinds of cells. So we have parietal cells, G cells, and chief cells. And there's a few smooth muscle cells kind of scattered around. So when they contract, it helps to kind of squirt the juice into the stomach. So we're going to first take a look at, notes here, the parietal cells. So the parietal cells secrete a couple different things. So one, they secrete intrinsic factor. You had a little bit of a rendezvous with intrinsic factor back in your article assignment in 232 when we had that guy with a gastrectomy and he had anemia, and what was his problem was, well, when you get your stomach taken out, you don't get intrinsic factor, and intrinsic factor is important for your body's absorption of vitamin B12. If you don't have vitamin B12, your red blood cells cannot mature properly and you get what's called pernicious anemia. So that was that connection back to our blood chapter. So this is where they're coming from. So the parietal cells secrete intrinsic factor. In addition, they secrete the hydrochloric acid. And I'm going to have a whole other slide on the next page to talk about the hydrochloric acid secretion, but it's really important for breaking down chemical bonds in the food that you're eating. It helps to activate the pepsinogen, which is going to come from another one of these glands. The acidity helps to kill bacteria, so we don't, the food that we eat is not sterile, so that acid helps to kill any pathogens, hopefully, the pathogens that are coming in. Yeah, okay. And then G cells produce hormones. They produce a hormone called gastrin, hence the name G for gastrin. And then chief cells secrete a hormone, a pre-hormone called pepsinogen. that then is activated in the presence of hydrochloric acid into pepsin. Pepsin does a job of breaking down proteins. So remember, proteins, big long chains of amino acids held together by peptide bonds. So pepsin breaks those bonds. It cleaves those peptide bonds, making the large polypeptides, the large proteins, and breaking them into smaller or shorter chains. It's not a complete digestion. It's just the beginning, the partial digestion of proteins, the beginning. That's going to finish the job in the small intestine. Okay, so let's take a closer look at the parietal cells. So these are the ones that are secreting hydrochloric acid. So this is a zoom way in to one of those cells lining the gland. So if you can orient yourself, we are in the lumen is on this side, interstitial fluid basically to blood is over here. They kind of show that right here. So we have a familiar chemical reaction, do we not? So we have carbon dioxide plus water goes to carbonic acid goes to hydrogen ions plus bicarb. All right, so now this is the second time we've seen this. We saw it in the respiratory system, we're seeing it in the digestive system, we're going to see it again in the kidneys, and we're going to see it again in the fluid electrolytes. So if you're still struggling with this concept of this buffer equation, you need to really kind of take a break and come and get some help, because this is vital to a lot of our processes in the digestive system as well as the kidneys. So hydrochloric acid, a big part of hydrochloric acid is hydrogen ions. A big part of any acid is hydrogen ions, because as you increase hydrogen ions, you decrease the pH. You make the situation more acidic. So let's take a look and see what happens. Get rid of all that clutter. Inside these parietal cells to make hydrochloric acid. So here we have our reactants, carbon dioxide and water, which is naturally found in our cytoplasm because our cells are making carbon dioxide. Our cytoplasm is mostly water. We have this enzyme called carbonic anhydrase, which speeds up that reaction. So you saw in the respiratory system, the little demo that I did where I bubbled my exhaled air through the phenol red, and it turned it from the neutral red color to the acidic yellow color. Well, it did it without carbonic anhydrase, and it took, some time to build up the carbon dioxide to change the pH. Carbonic anhydrase makes this reaction happen way, way fast. So we're not waiting for this conversion to the bicarb and the hydrogen ions. So here we're getting our products because carbonic acid quickly dissociates as an acid. It breaks off, releases a hydrogen ion. Now we want to take those hydrogen ions and we want those to go into the lumen of the gastric gland, because that's where we want the hydrogen ions to do their job. We're wanting them to be the acidic nature that's going to kill microbes, and the acidic nature that's going to activate pepsinogen, and the acidic nature that's going to break down connective tissues of plant and animal. And then we want to get rid of the bicarb, because that would buffer it. We don't want those two reattach, because if they were to reattach, we would like neutralize our acid. So we use active transport mechanisms to pump the hydrogen ions into the gastric gland. So you can see the yellow circles representing active transport. So what that means, we're going against the concentration gradient. All right, so that's our hydrogen. And now we got to get rid of our bicarb. Now, if you take a look, we have a counter transport mechanism where bicarb is getting pumped out, chlorine is getting pumped in. Now, does that look familiar to you, that little protein? It should, because that's a little protein that we saw in the respiratory system when we were transporting carbon dioxide from the tissues to the lungs. There, we call it the chloride shift. So we made the bicarb inside of the red blood cell, we pumped it out of the red blood cell to travel back to the lungs via the plasma as the bicarbonate ion, while the chlorine ion hitched a ride inside the red blood cell. And then when we got to the lungs, we kind of reversed that. So it's the same protein, which is cool. It's in our red blood cell membranes and it's in our parietal cell membranes. And so its job is to exchange the bicarb for the chlorine, and then the chlorine will just diffuse because it's high concentrations inside and will facilitate a diffusion because it's an ion, it can't freely diffuse, so it's got to go through a channel. And so here is our hydrochloric acid. So now that's going to be in the lumen of the gastric gland. It's going to go and be secreted and mix with all the chyme that your stomach is turning together. And then on the flip side of this, the bicarb goes into the bloodstream. Bicarb has the ability to tie up hydrogen ions, so it would be considered a base because it would reduce the concentrations of hydrogen ions. So when you eat a meal and your stomach is getting stimulated to secrete hydrochloric acid into the stomach, we get a little boost of bicarb going into the bloodstream, and we call this the alkaline tide. Okay, alkaline because it's basic. We're dumping a whole bunch of bicarb into the bloodstream. So if you were to take your blood's pH during digestion, right after you eat, you might see it go a tick up towards an alkaline range a little bit. That's because our stomach is pumping hydrogen ions into the stomach, or these parietal cells are pumping hydrogen ions into the stomach, pumping bicarb into your bloodstream. Now, when we take a look at our pH imbalances, when we get to chapter 27, one of the things that can cause a pH imbalance is vomiting. Repeatedly vomiting. So you're vomiting. You're going to be vomiting out all of your digestive juices. Your stomach's going to say, hey, we need to make more hydrochloric acid. So it's going to make more hydrochloric acid. You're going to vomit that out. It's going to make more, vomit that out. So what you're doing is while you're vomiting out your hydrochloric acid, you're dumping a whole bunch of bicarb into your blood. And this could lead to what we call metabolic alkalosis. And we'll see that more in chapter 27. So it's foreshadowing. So remember this lecture for when we get to chapter 27. Okay, so that's our hydrochloric acid production of the parietal cells using that carbonic acid bicarbonate buffer equation. We love that. Okay, so the last piece of the stomach is the regulation of gastric activity. There's 3 stages: the cephalic phase, the gastric phase, and the intestinal phase. So I'm going to work through each one, and then we'll see them all 3 at the end. So let's say you're just chilling. You've got your stomach. It's just relaxing. It's parasympathetic activating. It's just chilling. But you're hungry. Food's coming, but you're not eating yet. So the food isn't into your stomach yet. But your stomach is getting ready. So what's kind of cool, the reason why this is called the cephalic phase is because it is regulated by your central nervous system. Your conscious expectation of food actually preps your stomach for the arrival of food. So seeing food, right, that's your optic nerve. Smelling food, that's your olfactory nerve. Tasting food, that's your glossopharyngeal and facial nerves, right, for your taste buds. Or just thoughts of food, right, your hypothalamus, your limbic system, your emotional connection to food. So all of those things are processed in your higher centers. So your central nervous system is thinking about food, seeing food, smelling food, tasting food. All of those things are going to be sending action potentials down your vagus nerve. We can see that here with the red line, stimulatory. It's going to your submucosal plexus. Preganglionic neurons of a parasympathetic division. Postganglionic neurons will be going to all of these secretory cells. So your mucus cells are going to secrete mucus. Your chief cells are going to secrete pepsinogen. Your parietal cells are going to secrete hydrochloric acid. And your G cells actually secrete a hormone called gastrin. G for gastrin, that's right. So this is what's happening in that cephalic phase. It's very quick, about 5 minutes or so of this conscious stimulation of your stomach getting ready for food. It's almost like, you know, Pavlov's dogs, and he trained them to salivate at the sound of the bell. We don't have to be trained. Our stomach is salivating. It's doing all of these gastric juice secretions just by the thought or the smell or the taste or the sight of food. Okay, so let's say you're going to eat soon. You smell, you know, somebody's cooking dinner, and then dinner's served, and so then you consume the food. You chew, and then you swallow down to your esophagus, and now we have food in the stomach. Right, that's this yellow stuff. So we have a couple things going on here. So we have local control, because we're going to distend, we're going to feel, our stomach is going to feel that there's food in there. The pH is going to be changing. The chemicals showing up are going to be different. We have neural control because we stretch receptors, chemoreceptors are going to be activated. And we have endocrine control because that gastrin that was released in the cephalic phase is going to continue to be released in the gastric phase. So let's take a look at the neural control first. So distension. Empty stomach, full stomach. It's a stretchy bag. It's made of, the walls are made of smooth muscle. They have that elasticity, plasticity ability. So those are going to trigger stretch receptors. Stretch receptors are going to go to that submucosal plexus and the myenteric plexus. Submucosal plexus typically activates your mucus, chief, parietal, and G cells. So all of those secretory cells that got started in the cephalic phase, they're going to just be reinforced in the gastric phase. But now we're also communicating to the muscle. So it's churning. It's going to start mixing. So now we have the food that you've taken in as being secreted and mixing with more of these gastric secretions, and now physically mixing and churning together with those layers of the muscularis externa. Here's our elevated pH, right? So your stomach is typically a pH of 2. Hydrochloric acid, the gastric acid, is a pH of 2. When you take in food, your food that you're eating is typically not acidic. It's going to be more on the neutral side. So if you just ingest a whole bunch of neutral food, that's going to raise your pH, make it maybe 4, 5, or 6 pH. And that elevated pH can be picked up by these chemoreceptors that will do the same thing that the stretch receptors did. So submucosal and myenteric plexuses, activating mucus cells, chief cells, parietal cells, G cells, and mixing waves, because it's the plexus. You've just activated both plexuses. And this would be a short reflex, right? So these are short reflexes. So if we're going to talk about our visceral reflexes, the CNS is not a part of this anymore. This is a short reflex to the organ itself. And then lastly, so that's our local control, we have our short reflexes of the stretch and chemoreceptors, and now we have that gastrin. So gastrin was circulating in the cephalic phase. It's going to be secreted again by the G cells, and now it's coming back around, and gastrin, its target tissue, are the chief cells and the parietal cells, so we get those doubly with neural stimulation and hormonal stimulation to secrete more pepsinogen and hydrochloric acid. So you'll see those arrows are bigger than the mucus, because the mucus is just getting stimulated neurally, where chief and parietal cells are getting stimulated hormonally. So it takes hours. So your gastric phase is hours and hours of churning, depending on the size of the meal, obviously. So a granola bar is not going to take as long as a full course meal. Okay, so that's a gastric phase. Mixing, 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 churning, churning, churning. The carbohydrates started getting digested in the oral cavity with amylase. The proteins are starting to get digested in the stomach with a pepsin. The hydrochloric acid is breaking down cellulose of plant material and connective tissue of animal material, and it's turning into that chyme slurry. And that might take, 4 to 5, 2, 3, 4, 5 hours, depending on the size of your meal. Then we get to the intestinal phase. And what I want you to notice here is in the previous slide, almost all of the arrows were red or stimulatory. In the intestinal phase, all of the arrows are the blue inhibitory phases, because in the intestinal phase of gastric activity, we are slowing down the stomach because the food chyme is now starting to go into the intestines. And we don't want to keep churning and mixing the stomach if everything's ready to start moving into the small intestine. All right, so the trigger of the intestinal phase is stuff coming into the duodenum, which is this tube right here. It's the first part of the small intestine. I'm going to post a video of showing this pyloric valve. The music is horrible, but the imagery is really good. So it's showing the peristalsis of the stomach as it's letting a little bit of chyme go through the pyloric sphincter into the duodenum a little bit at a time. So what that's going to do is going to trigger stretch receptors in the duodenum and chemoreceptors. Stretch receptors because now there's actually chyme coming in, and chemoreceptors because it's low pH. It's like a pH of 2. And so that's going to come in, trigger these chemoreceptors and the stretch receptors, which will go and inhibit the myenteric plexus. And remember, the myenteric plexus was primarily the churning part. So the churning is going to slow down. It's not going to completely stop, but it's going to be inhibited. It's going to slow down. Okay. All right, so that's going, I'll do this little guy. So I should do it in blue, but there's your slowing of your peristalsis via the myenteric plexus. And that also is going to slow down any secretions. It's not shown here, but you'll notice that the secretions are also going to be inhibited. The other thing that's going to be triggered to be released, which we'll see in our hormonal mini lesson a little bit later, is the presence of lipids and carbs and this low pH. So we've got these chemoreceptors being able to recognize fats and sugars and an acidic pH trigger these three hormones. We have CCK, stands for cholecystokinin. We have GIP, stands for gastric inhibitory peptide, and we have secretin. So all three of those hormones gets released from the duodenum, these endocrine cells embedded in the wall of the duodenum, goes into the bloodstream, comes over and blocks chief cells, blocks parietal cells, and blocks peristalsis. So everything that we just turned on in gastric phase, we are turning off in the intestinal phase. We're slowing it down, and what that will do is it will allow the stomach to slowly release a little bit of chyme at a time, so it's not overwhelming the rest of the digestive process. We don't want to dump a whole stomach's worth of chyme into the small intestine at once. It wouldn't be properly processed. All right, so if we look at all of these three together, here we have our cephalic phase, which is the controlled by your brain, basically, your conscious thoughts and awareness. Gastric phase is all excitatory and stimulatory of your glands and your churning. And then intestinal phase is once it's all been churned and it's ready to go into the intestines send the signal to the stomach to slow down. And that's what this guy here is called, the enterogastric reflex. There's a couple of the reflexes I'll point out. Enterogastric means you're going from the intestines to the stomach to tell it to slow down. It's this neural built-in reflex, one of those short reflexes of gastric regulation. Okay, the last slide I'm going to show you, so I'm done. So this is all of the physiology of the stomach. But in class, I talk a little bit about this historical story. We usually listen to a podcast, but I couldn't find it, so I can't post it. But the story goes, there's this guy named Alexis St. Martin. That's him right here. And here's a doctor named William Beaumont. They were in Canada or northern parts of the United States in the 1800s. It was like a fur trading camp. A gun accident happened where Alexis St. Martin dropped his gun, or somehow a gun went off and it shot him right here in kind of his side, right under his diaphragm. And so he was taken, and William Beaumont was the doctor at the camp, and so he treated him, but he realized his wound was not healing. It was not closing up. And after a while, he made some observations that this fistula, this unhealed wound, was going directly into this man's stomach. And at this point in time, people didn't really understand digestion. They didn't know how the food that you eat turned into the nutrients and the energy that you needed. It was still a big mystery. So there's some controversy here as to whether Alexis consented to these experiments, but experiments were done. And basically what he would do, he would get food bits, all different kinds of food, tie it to an end of a string, lower the food bit into the stomach, let it sit, pull it out, take notes of the varied stages of digestion of the food item, and put it back in and let it continue to digest. And he'd pull it out so repeatedly. And so he came up with how long it would take to digest certain food items like eggs or meat or cheese or vegetables or whatever he was testing. He also siphoned out some of the acid, which looks like what he's doing there. He tasted it. He did other experiments with the gastric juice, like in flasks and stuff outside of the stomach, because again, they didn't know what caused digestion. It was accepted that it was just kind of some magical event that happened, but with William Beaumont's experiments with this unfortunate Alexis St. Martin, he concluded that it wasn't anything magical. It was these chemicals that are inside of the stomach that digest the food, because he could get the same type of results if he extracted some of the acid and put the food in a beaker outside of the human body. So his research actually gave a lot of knowledge about the stomach and the digestive system to the scientific and medical community, even though his ways of gaining that knowledge wasn't quite ethical. And then there was some back and forth of he was working for him. Alexis was working for Dr. Beaumont, but didn't like being the guinea pig anymore and left, but then he came back. And the end of the story is Alexis St. Martin lived for quite some time, had a family, but eventually died. And his family did not want his body to go to science or anything like that. So the story goes, they left his body out to rot for days and then buried it way deeper than any grave normally was at that time, because they didn't want anybody to come and exhume his body and do more research on him. So kind of a sad ending to this experimenting lifestyle. Anyways, look it up if you're interested in the Alexis St. Martin and William Beaumont relationship, I guess you can call it. Very interesting stuff. Okay. All right, that's it for the stomach. I will see you next time.